Hello, and thank you for joining me today at LabRoots. My name is Kyle Nilson, and I'm from QIAGEN Digital Insights, where we work to make sure you have access to the best expertly curated data there is. Today, I want to go ahead and talk about both our own data as well as the curated data from our partners like Cosmic, where we can use this information to try to avoid pitfalls when performing drug discovery and to also use this in order to get a better understanding of exactly what mechanisms of action are occurring or are relevant to your given therapy of interest. Although the use cases that I'm showing today are intended for research use only, it is possible to license these things for clinical diagnostic purposes. So talk with your QIAGEN rep. Also, as a heads up, a license is required to use either Cosmic or HGMD for any kind of commercial or clinical use. And this does include the website. Finally, if you are under license, I am available to help you out. Our tech support is always available here at ts-bioinformatics at QIAGEN.com. But in addition, I love helping people out with their use cases. So if you have some sort of discovery research need, go ahead and let me know, because follow-up assistance, including one-on-one help, is included, again, with your license. So today, I want to talk about two use cases. One of them is going to take advantage of the huge breadth available within Cosmic to look for any kind of mutational evidence. For example, let's say you're developing some sort of gene therapy. If you are aware of any kind of off-target effects from, let's say, a CRISPR knockout or edit, it is imperative to evaluate whether or not those effects could lead to any kind of significant cancer or other deleterious effects. Something that Cosmic is fantastic for is it contains the largest, most comprehensive database of any kind of somatic mutations. So this is across all genes and across all cancer. And it's possible to use this not only to look, let's say, in a genome browser using a web interface and evaluate certain genes or mutational hotspots, but if you license Cosmic from QIAGEN, you also get access to the cancer mutation census, as well as actionability information that can let you prioritize relevant variants and avoid anything that is known to cause cancer. The second use case that I'd like to talk about is instead performing more of a deep dive. And in this case, this is starting with a use case in TCGA, where because of the immune landscape of cancer, we now know that host immune response plays a significant role in survival and prognosis. It turns out that if you carefully consider differences in host immune responses to low-grade glioma, you can find a network of genes that changes based on the most clinically common variant in this cancer. And by mapping this out using QIAGEN resources, it is possible to see how this can lead to a decrease in survival, but also possible co-therapies that could theoretically be developed in your lab. So in order to cover those use cases, I want to go ahead and first cover what I mean by expert-curated content. And this will include just an overview of the research findings, omics data, and gene variants that both QIAGEN and our partners put together. And also just a brief discussion about how they can be used together. After that, we will focus on those two distinct use cases, one of which is going to broadly search mutations in Cosmic in order to get an assessment across all sorts of genomic locations to see whether or not there are deleterious mutational effects from let's say, your gene therapy and development. The second use case, again, is where we will go ahead and deep dive into specific cancer details. In this case, low-grade glioma, taking advantage of the curated TCGA data within OmicsSoft, as well as HSMD, which is a fantastic resource for clinical frequency of somatic variants. But again, first, just that quick introduction. So why do we insist on expert curation of public data? It turns out that it is absolutely essential to have scientific cleanup and scientific interpretation of the results. So about a third of the time, there's usually some sort of mistake that will not be captured by any kind of machine learning or language model ingestion of data. A perfect example actually is from my own graduate work, where if I performed K562 chip sequencing and I uploaded my metadata table to Geo, I unfortunately dragged and dropped in Excel and had K563, K564, K565 cells. Any scientist can immediately recognize and fix that error, but that will go completely unnoticed if you are using automated data ingestion. And the only way to pick those details out is if a scientist read and input the data in the first place. So we apply that effort both for the creation of our databases, but also for the interpretation of findings. We make sure that we have at least 2 scientists review every fact before it enters our various collections. We provide a large variety of different types of data, both our own data as well as partner data from the Sanger Institute for Cosmic, as well as from HGMD Professional, a fantastic germline resource for those looking in rare disease. The 3 types of findings that I want to discuss today are, first, relationships. When you consider how different genes or proteins interact with each other, there will inevitably be some sort of node, relationship node type of effect, where protein A might phosphorylate protein B, leading to an increase in activity. All of those details are captured by our team of curators for use in the QIAGEN biomedical knowledge base. And that knowledge base is what powers Ingenuity Pathway Analysis, a fantastic relationship resource that has been growing over the past 20 years. Next, I want to talk about omics data. Although the relationships that are present in Ingenuity Pathway Analysis are incredibly useful for coming up with really cool mechanisms of action, sometimes you need that real-world context. And in this case, this comes from hundreds of thousands of individual data sets that contain RNA-seq, microarray data, DNA sequencing, mass spec, other proteomic assays. And all of this data has been processed by our scientists so that it is normalized and all uses controlled vocabulary. In other words, we make sure that all of the treatments use a unified term, all of the diseases use the same ontology, and everything can be cross-compared. This means that it's possible to look at a very large number of samples across different experiments to find effects such as differences in expression due to mutation, or differences that are also due to, let's say, host immune response. Finally, curated gene variants are an ever-growing and important part of research. And with that, we provide our own human somatic mutation database, which takes advantage of the real-world clinical information we have from N of 1. But we also partner with the Sanger Institute for Cosmic, so that way we have incredible breadth as well as incredible depth to offer. Just going over the different types of data. Again, the relationships that are present within ingenuity pathway analysis are kept up to date. We make sure those relationships are updated from journal articles on a weekly basis. If you are using the biomedical knowledge base more directly, that is updated quarterly. And ultimately, millions of findings go into this underlying knowledge graph where we take any kind of curated experiment where we can see how two different proteins might affect each other, or how a drug might lead to inhibition of a protein, and how that can affect downstream diseases. And ultimately, all of that information is provided to scientists through ingenuity pathway analysis, our graphical user interface, and an application that we've had for over 20 years. We also have recently started offering Python and R API access, as well as flat file access to the underlying data. So if you happen to be interested, you should let us know and find out more about this particular offering. The omics data that we have, this is where we have a few different key elements. In addition to curating projects based on customer requests, we then have to make sure that we clean up and quality control the data in terms of metadata, as well as the actual sequencing information. So for metadata curation, that means we are making sure that there aren't any kind of mistakes and that everything has a unified vocabulary. So that means remapping everything to our own ontologies, and then also taking the time to make sure that those changes or any kind of curation we do make sense and match the original manuscript as well as any other conclusions in the field. The data processing is where, for RNA sequencing and microarray, we actually reprocess the data from scratch. And because we are reanalyzing the data, we can normalize the data to a 70th percentile, for example, and then from there, we can go ahead and perform both quality control, again, looking for swap samples or any kind of mistake, but also statistical analyses. So for every experiment, if a study has case and control conditions, let's say disease versus normal, or tumor versus wild type, or even things like treatment versus no treatment, sensitive versus resistant, etc. All of those comparisons are done on a study-by-study basis. We calculate the differential gene expression or differential protein level for each study, and we store all of the calculated log2 fold changes as well as adjusted and raw p-values. So that way, instead of searching for all instances of how a gene might be expressed, you can instead search for all instances where a gene might be significantly up or down and see what those conditions are. And all of that information, again, we try to make available as many ways as possible. And that can be through our user interface, Omics Studio, or the website Land Explorer, or our recent Python and R offerings for accessing this by API. The final type of data that I want to talk about is variant data. And COSMIC continues to be the world's most comprehensive gold standard reference database when you're looking for any kind of cancer annotation. COSMIC version 99 came out at the end of November. And in this case, we are very close to 25 million total genomic variants in this database. And all of that information covers a breadth of sources. However, we like to pair that with our own human somatic mutation database. And in this case, one of the things that we provide is clinical depth. So COSMIC, because it has so many variants, it can sometimes be difficult in order to figure out which variants are the most frequent in a given cancer type. Or you might be interested in some other clinical details. HSMD was built from the ground up using clinical data. And because of that, we have millions of variants over a very small number of genes. And all of that information is based on over 500,000 clinical oncology use cases. So something that I like advocating is taking advantage of all the data that you have. So for COSMIC and HSMD, it could make sense to use COSMIC for your initial screening and then HSMD in order to dig further in depth. Likewise, for our other information, I'm a big fan of using our knowledge base or ingenuity pathway analysis to come up with big questions and answer broad hypotheses. But then I will then use the genomic information present in Omicssoft to determine whether or not my prospective candidates are in fact differentially expressed in specific tissues, diseases, or cell types. So for this initial use case, I want to go ahead and show you how you can take advantage of COSMIC to broadly search for any kind of evidence of mutational effects. So for this use case, let's say that you're interested in developing a CRISPR therapeutic. And in order for that to pass muster, you need to confirm that any changes that you are making to the genome will not have a deleterious effect. One of the ways to do that is to search the most comprehensive mutation database related to cancer, COSMIC. We have COSMIC available in a variety of different ways, and I'd like to show just what some of these are. For example, COSMIC data can be browsed on their website. And when looking at this type of resource, it is possible to go ahead and say, look at a specific genomic region of interest. So here, I'm just copying and pasting a particular loci where I might have experimental or predicted evidence suggesting that I might have an off-target effect in this region. So here on COSMIC's website, I can very easily choose a genomic region and search for any annotated variants that might be present in that region. And here you can see that for the 500 base pair window that I selected, there happens to be a large number of annotated variants. So at face value, this might be a region to possibly avoid, especially when, if you consider scrolling out, you'll see that other regions in the neighboring area are not mutational hotspots. However, if you look in more detail at some of these, it turns out that COSMIC also has a number of incredibly handy resources. For example, the cancer mutation and cancer gene census, as well as actionability information. So the cancer mutation census can help you find specific information about a particular gene. So for example, here when we were looking at these variants, if I happen to open one of them, I can see that we are looking at this CAL-HM3 gene. The cancer mutation census, the website does require you to create an account. So this does, again, require a license. However, if you do get that license, not only can you use the website, you can also take advantage of direct access. So here on QIAGEN's website, I'm showing that it's possible for commercial users to download the data underlying Cosmic. And while this loads, here I'll show that you have comprehensive release notes, as well as very detailed flat files for all of the cell line project information, all of the individual genes and mutations that are tracked within Cosmic, as well as access to the mutation census information, the gene census information, and the actionability information. So all of this can be found on the QIAGEN website, as well as by looking at the Cosmic website for more information. That said, it can be a little daunting to look at this and realize that you have a huge amount of flat files to parse through. Fortunately, that's where programmatic access can really help you out. And this makes it possible for you to integrate Cosmic data into your own analysis pipelines. For example, here, I'm showing you a somewhat crude shell script that I cooked up that can search around a given genomic location. And once I search around this location, what my little application will do is it will take all of the known variants within that genomic window and report them, whether they are coding or non-coding. And then from there, I will look in the cancer mutation census for all of those variants to see whether or not any of these have clinical or prognostic relevance. So here, that's just a really quick awk script. If I go ahead and switch to the applet, this is where I can show that by looking at the particular region of interest, so here again, I am looking 10 base pairs around this particular location on chromosome 10. I can see that there are, in fact, a variety of visible annotations. So here, if I scroll up, sorry for accidentally clicking and scrolling down, you can see that we have a number of annotated variants. And for these variants, it is possible to then look in more depth to see if there's any kind of cancer mutation census information for those genes. So this is where I can find out what gene this is, and also what are the specific details about the mutation, what are the genomic locations in HG37 as well as HG38, and also if there is known mutational significance, or if we have clear-cut mechanistic links between those genes and cancer. And finally, if we happen to see any kind of frequency for a specific cancer type within all of the Cosmic samples, this is something that we can also pull out. So here, just scrolling through, I can see at a glance that all of these variants have no clear-cut consequence for causing cancer. And if necessary, it's possible to also follow up with all of the manuscripts and see whether or not any of those have additional information. But in addition to these specific windows, again, because it is possible to look in other genomic regions, you can determine fairly easily whether or not there are different locations that might be more ideal. So for example, if I look 20,000 base pairs away, I can go ahead and search for this new window just to see how many variants are present here. And so this might take a second, but ultimately, the main thing to point out about this is you have the ability to look at all of the variants within a given region programmatically, and you can easily pull out information related to ClinVar, nomad significance. You have access to expert interpretations for whether or not these are causal variants. And you also have access to actionability data, which can help link a given mutation to known drugs as well as known specific cancer types. And just peeking over to see if this is done. Unfortunately not. So moving on to the second particular use case, what if you want to dive a little bit more in depth? This is where we can really take advantage of the human somatic mutation database that QIAGEN offers, as well as our curated omics data and our curated relationships. So for this particular use case, I want to focus on some of the findings present within the TCGA marker papers. And from there, tease out a mechanism for why low-grade glioma seems to potentially be affected so much by host immune response. So just in terms of background, cancer outcome is influenced by tumor microenvironment, as well as by how the host responds to that cancer. And the Cancer Genome Consortia took the time to annotate and characterize these host immune responses in a series of papers that came out in 2018. In these manuscripts, they described 6 particular responses. And in addition to finding these, they also annotated a very large number of their existing samples. And it turns out that at a glance, we can see that low-grade glioma has a really interesting response to this cancer. So whereas most cancers have some mixture of wound healing, interferon gamma dominant, or inflammatory responses, it turns out that for low-grade glioma, the responses tend to either be from this cluster number 4, which is lymphocyte depleted, or this cluster number 5, which is immunologically quiet. And it turns out that if we look within TCGA data that has been curated, we can see clearly that this split affects survival for low-grade glioma. The final piece of the puzzle is the list of genes and proteins that TCGA found to form a pan-cancer regulatory network to influence this host immune response. And in particular, the authors grouped these genes as somatic mutation targets, transcription factors, and immunomodulators. And so at this point, we need to build a potential case for what might be going on. And because cancer is so significantly influenced by mutation status, this is where I personally wanted to look in depth at the cancer mutation status of these low-grade gliomas. And to get that information, again, I went to the QIAGEN website and logged in to HSMD. HSMD is built off of a huge number of clinical cases. And because of this, you can search across genes, diseases, variants. And if I type in LGG for low-grade glioma, this is where I can get clinical case distributions for this particular cancer. So scrolling down, I can see very clearly what genes are most frequently mutated in these cancers. And low-grade glioma, in this case, surprisingly, it's not TP53, but in fact, IDH1 is the most frequently mutated gene. And in addition to this specific gene, scrolling down, it is also possible to look at the specific variants, the alterations present. And this is where overwhelmingly the most common mutation that appears in these cancers is IDH1 R132H. So I already have a variant of interest. And if I want to search for this, I can very easily pull up this particular variant. So here, IDH1, R132H. Looking at this particular variant, it's a missense alteration. It's within a hotspot. And it also has top tier actionability and clinical significance. And this is where I can find a lot of information about clinical trials and drug association. So here, scrolling down, this is where, if needed, I can look at the various distributions of allele frequency, and I can also see what drugs are possibly influenced. And ultimately, all of these findings are summarized in very clear language by our expert curation team. So here, this is where it's very easy to digest and get to the information that you need. So shifting back to this overall use case and need for discovery, this is where I've set the stage to focus on low-grade glioma. Because of TCGA, I know already that I'm interested in looking at cluster 4 versus cluster 5 host immune response. And now, thanks to HSMD, I now also know that I want to focus on IDH1 wild type versus the most common clinically relevant variant, R132H. And if I go ahead and analyze this data, this is where I can take advantage of all of our curated expression data within OmicsSoft Studio and our OmicsSoft lands. In this particular case, I'm looking at all of the TCGA samples that have known mutation information for IDH1, as well as known host immune response to the various cancers. And with that, I went ahead and searched for any kind of significant difference in any gene between cluster 4, that lymphocyte depleted immune response, and cluster 5, that immunologically quiet response, as well as any difference between mutant and wild type for IDH1 R132H. And here, it turns out that there is a significant change in ETV7 expression when you mutate IDH1. And this change is much more pronounced in cluster 4. So here, by looking at this heat map, it is in fact possible to perhaps tease that out by the naked eye. But instead, something that I like pointing out is that you can also use a programmatic or big data approach to ask for all of the genes that have some sort of differential expression based on whether or not you are mutant or wild type, as well as whether or not you are in cluster 4 or cluster 5. And so here, if I go ahead and search for ETV7, this is where I can show what I mean by omics data and how it's curated. So just through a simple search, this is where I can pull up hundreds, if not thousands, of sequencing samples and look at the distribution of this particular gene. So one of the first things that I'll do is I'm looking at all TCGA samples, except I went ahead and already grouped samples based on whether or not they are part of this cluster 4 or cluster 5 response, and whether or not they possess this mutation. So if I go ahead and look at the RNA-seq gene expression information for ETV7, this is where I can go ahead and, let's say, look at this data, and I can also split this data out based on this custom query, where I'm looking at IDH1 mutation status. So now I have two charts, which I'm going to just lay on top of each other. And this is where I can see that I have both wild type gene expression for R132H. Excuse me, the expression is of ETV7 and the wild type protein is IDH1, or I have expression of ETV7 when IDH1 is mutated. And when I show this in a pairwise fashion, I can see that both of them show significant changes in expression, though that change is much more pronounced when considering the immune response for these cancers. So here, to specifically answer that, I would want to go ahead and change how I have these samples grouped. And then when I split these charts, this is where I can go ahead and split them based on host immune response. So here, searching for immune metadata, I am adding this as a potential trellis option. And then I can easily sort these by p-value and show the summary information. The final piece of the puzzle is, how is this impacting survival? How is this relevant for drug discovery? And to address that, this is where I turned to the relationships contained within our biomedical KB. So for this particular example, I took all of the genes from the TCGA manuscripts related to host immune response. And I also wanted to look at, say, common drug targets for treating low-grade glioma. And in this case, I happen to focus on bevacizumab. Again, because we have relationship information, I can see clearly that when I increase bevacizumab or simulate an increase of bevacizumab, that will normally lead to a change of activity, which is either going to be an increase of certain gene activities or a decrease of those activities. So here, when bevacizumab is up, this knowledge graph is predicting that VEGF should be inhibited, and through this network should ultimately lead to a decrease in low-grade glioma disease activity. However, this is where we can see a potential issue. If we instead simulate a decrease in ETV7, which is what we see in the poor survival cohorts for low-grade glioma, this in turn is leading to activation of bevacizumab targets and VEGF receptor. So at this point, it's just a matter of simplifying the network and trying to identify ways to tease out this particular network and come up with a potential mechanism that leads to a good outcome. So for these particular samples where you might have theoretically decreased ETV expression due to this mutation, a hypothetical activator could restore typical VEGF behavior and thus improve responsiveness to bevacizumab. However, that is a hypothetical. If we instead look through our relationships for known drugs, it turns out there is a known inhibitor of ICAM1, the target immediately downstream of ETV7. And when simulating an increase of that tripeptide, that led to overcoming the particular effects on VEGF receptor, thus restoring bevacizumab responsiveness. Again, this is all looking at hypothetical behavior based on literature connections. So ultimately, it would be up to you to look in the lab to see whether or not that's actually true and what other compounds could be used as a potential co-treatment in these low-grade gliomas with otherwise poor survival outcome. Just to summarize all of the different curated data options that I showed today and how you can possibly use them for your own drug discovery, I showed one way that you can look broadly within COSMIC, look broadly at somatic mutation evidence in order to determine if you might have off-target effects that could be very poor for outcome. So this can let you prioritize and ultimately avoid projects where you would otherwise not get regulatory approval due to these potential risks. In addition, I also showed a way that you can use some of the deeper knowledge contained within our databases, where I used HSMD in order to identify the clinical frequency of various mutations in low-grade glioma. And once I looked at the most common, the one with the largest market size, I was then able to look at our curated omics data to identify exactly which transcription factors had the biggest change due to this mutation or due to the host immune response. And then finally, I used our biomedical knowledge base in order to identify a network and simulate a mechanism of action that indicates potential ways to overcome this deleterious effect. And again, if I want to have one big take-home message, it's that using both of these resources hand in hand can really help make a difference, because you need to have breadth in order to survey a huge amount of mutational information, a huge number of potential genomic loci. But you also want to have real-world clinical data in order to get all of the details nailed down. So with that, thank you again for attending LabRoots, and please don't hesitate to reach out if you have any questions whatsoever, or you want to explore your own use cases with me. I'm more than happy to schedule some sort of one-on-one training or just a brainstorming session. Have a great day.